Hi, I'm Dodie Sanders with the University of Georgia Marine Extension and Georgia Sea Grant. We're going to be covering in Module 2 on microplastics, the standard operating procedure for the collection and visual identification of microplastics in water samples. So here are some of the field guidelines for collection that we always follow, and it's a series of steps, a protocol to ensure maintaining sampling consistency and to eliminate contamination of the samples. So for instance, we sample at the same site, recording the GPS coordinates, the latitude and the longitude. We collect the water samples at about the same tidal stage, always during an outgoing tide. We always wear cotton clothing, and this reduces the likelihood of contaminating the samples. We work downwind during collection process, and we record the conditions at the site, including the weather and the tide stage. We also use 40 ml glass vials that have been combusted at 400 degrees Celsius for at least 5 hours. And along with these vials, we have pre-cleaned lids that we've washed and sonicated for 15 minutes in a 0.2 micron filtered deionized water bath. And we also rinse those lids again with a 0.2 micron filtered deionized water. The sample collection protocol for each site also involves labeling in advance the vials that we're going to use for the samples. And so the label vials have the corresponding site name, the stainless steel sieve size, the replicate number, and the date. And you can see here that we have a series of sieve size ranging from 355 micron to 106 to 63 micron. And we also use 3 blanks. These are vials with deionized water for each site, and a sample blank using the 63 micron sieve, 1 for each site. At the site, we rinse each sieve well with 0.2 micron filtered deionized water, and then we stack the sieves with the 355 micron sieve on the top, following the 106 in the middle, and the 63 micron on the bottom. When we get to the site, we use a stainless steel bucket that has been marked at 4 liters on the inside. We stand downwind of the stainless steel bucket. We rinse the bucket with the site water 3 times, and then on the 4th collection, we fill the bucket to the 4-liter mark, again, located on the inside of the bucket. And that really ensures that we collect the same volume at each site. We pour the 4 liters of water through the sieves, and it's really helpful to have one person holding the sieves while a second person pours the water from the bucket. We then separate the sieves, making sure to keep them upright. And using the squirt bottle with filtered deionized water, we hold the sieve at about a 45-degree angle so that we can rinse the inside of the sieve while moving the particles from the inside top of the sieve to the inside lower lip. And then we rinse a metal funnel with deionized water. We place that rinsed metal funnel in a vial. And then we rinse the sample from the inside lower lip of that stainless steel sieve into the funnel, and eventually the sample gets into the vial. With the funnel still in the vial, we rinse the funnel with deionized water to ensure that nothing remains in the funnel. And then we rinse a cap, again with deionized water, and we screw on the vial and place the sample in a test tube rack for storage. And we repeat rinsing steps with the 2 remaining sieves, 3 replicates for each size class of sieve at each site. We also do sample blanks, and this is using another labeled vial, the site name, sample name, and the date. And we rinse the 63 micron sieve with deionized water, and we use the same protocol as the sample collection for the other sieves. We also do filtered deionized water blanks, and we fill 3 labeled vials, again labeled the same way as the other samples, with filtered deionized water from the squirt bottle. We rinse the lids, we put them on the vials and place those vials in the test tube rack. And then for multiple stations, we always rinse the sieves and funnels thoroughly with the 0.2 micron filtered deionized water. We cover and store in a clean container with a lid between stations. We store the field book, the field log book, the samples and the blanks together in a dark, cool place until we're able to analyze them in the lab. And keeping in mind with the sample collection and the processing and the analysis, here's some things that we always keep in mind. We combust all the glassware used in the filtration of the samples. And then prior to filtering the samples onto a 47 millimeter, 3 micron cellulose acetate filter, the filters and the petri dishes that the filters are stored in are always visually inspected for contamination using a microscope. And this is done in advance. Most important, we use a laminar flow hood for all filtration and filter handling while wearing cotton clothing like a cotton lab coat. Sample filters are archived in a sealed, pre-cleaned, and visually inspected plastic petri dish. And again, when we do the visual inspection for microplastics, we're really using morphological features. We're using the size, the shape, and the color. We're not at this stage able to detect, or using this method, be able to detect the polymer or plastic type. But if you're interested in just an introduction to microplastic identification, here are a couple of sources that we've used in the past. So I just want to remind you that we do have a couple of other modules, Module 1 on microplastics and introduction, and Module 3, the distribution of plastics and looking at the potential impacts. So that concludes the second module to the microplastics, kind of the protocol and the procedures. Thanks very much.